This is the most expensive liquid on the planet, and it isn't oil, perfume, or printer ink. It is blood. Bright blue blood drawn from an animal older than the dinosaurs. 1 quart of this processed blood sells for $60,000, making it worth 15 times more than gold. Every vaccine injected into your arm, every surgical implant placed inside your body, must first pass a test using this blood. The horseshoe crab has survived 450 million years of extinction events, and now it saves human lives. We'll follow every step of the journey, from ocean floor to pharmaceutical laboratory. The process begins before sunrise on the Atlantic coast, where licensed harvesting crews prepare for a carefully regulated catch. Delaware Bay is the epicenter, home to the largest spawning population of horseshoe crabs anywhere on Earth. Licensed crews use modified trawl nets designed to capture the crabs without damaging their shells or limbs. Each crab is handled individually. Any animal showing signs of injury or stress gets returned immediately to the water. A single boat can bring in several hundred crabs per trip, but strict quotas cap the total harvest. The crabs are loaded into ventilated containers kept damp with seawater. Temperature and moisture must be maintained throughout transport. From dock to laboratory, the entire journey takes less than 24 hours. Speed is critical to keeping the crabs healthy. Refrigerated trucks rush the harvest to one of only 5 licensed bleeding facilities in the entire United States. These highly regulated facilities operate under strict federal and state permits, each one inspected multiple times per year. When the truck arrives at the bleeding facility, the clock starts. Every minute counts from this point forward. Technicians wearing full protective equipment transfer the crabs into the sterile receiving area for immediate processing. Every single crab undergoes a health inspection. Size, shell condition, and overall vitality are all checked before bleeding begins. Technicians look for healthy gill movement and responsive limbs, signs that the animal can safely tolerate the extraction process. The crabs are positioned on specially designed stainless steel racks that hold them securely at a downward angle, exposing the cardiac region. Inside the bleeding room, hundreds of crabs line up in rows, each one connected to its own sterile collection vessel. A sterile needle is inserted near the heart, and within seconds, that unmistakable blue blood begins to flow. The blood flows entirely by gravity. No pumps, no suction. Just the crab's own circulatory pressure filling each bottle drop by drop. That stunning blue color comes from copper. Horseshoe crab blood uses copper-based hemocyanin instead of the iron-based hemoglobin in our own veins. Each crab gives roughly 30% of its blood volume over a controlled extraction window, carefully timed to protect the animal. When the timer signals completion, the needle is carefully removed, and the puncture site is left to clot naturally. After extraction, every crab enters a monitored recovery tank, resting in circulating seawater before the journey back to the ocean. Within 48 hours of capture, the bled crabs are transported back to the exact coastline where they were collected. The return rate is strictly tracked. Every crab that leaves the facility must be accounted for and released at its original harvest site. This catch, bleed, release cycle has continued for over 50 years, the foundation of a multi-billion-dollar pharmaceutical safety industry. Back inside the laboratory, the real transformation begins, turning raw blue blood into one of medicine's most critical testing reagents. The collected blood is first filtered to remove any debris, then transferred into high-speed centrifuge tubes for separation. The centrifuge spins at thousands of revolutions per minute, separating the blood into distinct layers within minutes. After spinning, the blood separates cleanly. The plasma rises to the top, while the precious amebocyte cells settle into a dense pellet at the bottom. The plasma is discarded. It is the amebocyte cell pellet that contains the clotting factor worth $60,000 per quart. The isolated cells are resuspended in a sterile buffer solution. This prepares them for the critical lysis step that breaks them open. Under a microscope, these amebocytes are remarkable. each one packed with granules containing the clotting enzyme that detects deadly bacteria. The lysis process ruptures every cell membrane, releasing the clotting factors into solution and creating the crude lysate. The crude lysate passes through a cascade of precision filters, each one removing cellular debris while preserving the active clotting proteins. Chromatography columns further purify the extract, isolating the specific proteins responsible for the bacterial detection reaction. Drop by drop, the purified Limulus amebocyte lysate collects in sterile flasks, the active ingredient that will protect millions of patients. This is Limulus amebocyte lysate, named after the horseshoe crab genus Limulus and the cells that produce it. The purified lysate is dispensed into thousands of individual test vials, each one containing exactly the right concentration for a single endotoxin test. The filled vials enter industrial freeze dryers, machines that remove all moisture while preserving the delicate protein structure intact. Over 48 hours of freeze drying, the liquid transforms into a stable white powder with a shelf life measured in years rather than days. Automated systems seal each vial with pharmaceutical-grade stoppers, ensuring no moisture or contaminants can reach the reagent inside. Before a single vial leaves this facility, it must pass over a dozen quality control checks, each one documented and traceable. The gold standard test is simple but elegant. If the LAL detects bacterial endotoxins, it forms a solid gel clot within 60 minutes. A positive result means contamination is present. That batch of medicine gets rejected before it ever reaches a patient. Every test result is logged into a traceable database, linking each vial of LAL back to the specific batch of crabs that produced it. Environmental controls monitor every room in the facility. Temperature swings of even 2 degrees can destroy an entire production batch. Positive pressure airlocks separate each processing zone, ensuring contaminants from the outside world never reach the product. Each production batch undergoes sensitivity testing, confirming the LAL can detect endotoxin concentrations as low as 0.03 units per milliliter. That sensitivity is extraordinary. LAL can detect quantities of bacterial contamination invisible to virtually every other testing method available. Modern facilities use kinetic turbodimetric readers that can process hundreds of samples per H-hour, measuring gel formation speed with laser precision. 96 samples tested simultaneously. The reaction time in each well reveals the exact concentration of endotoxins present. Across the world, pharmaceutical companies rely on this reagent to certify every injectable product they manufacture as safe for human use. The FDA mandates endotoxin testing for all injectable drugs, vaccines, and implantable medical devices. There are no exceptions. Every bag of IV saline, every vial of insulin, every dose of chemotherapy, all must test negative for endotoxins before reaching patients. A single LAL test kit costs between $200 and $1,000, and global demand consumes over 70 million tests every year. The global LAL market generates over $1 billion annually, all of it traceable back to the blue blood of horseshoe crabs. Finished LAL kits ship worldwide in temperature-controlled packaging. The reagent must stay cold from factory floor to pharmaceutical lab. From this single facility, shipments reach pharmaceutical companies in over 40 countries. A global supply chain built on ancient biology. North America, Europe, and Asia consume 95% of the world's LAL supply, with demand growing every year. During the pandemic, Louisiana L testing became front-page news. Every single dose of every approved vaccine required endotoxin certification. Billions of vaccine doses meant billions of additional LAL tests, pushing production facilities to their absolute limits. Each vaccine batch requires multiple tests at different production stages. A single failure sends the entire batch back for reprocessing. But horseshoe crab blood doesn't just protect drugs. it is also advancing the cutting edge of biomedical research. Researchers use LAL to study bacterial infections, develop new antibiotics, and test experimental treatments long before human trials begin. Every artificial hip, every pacemaker, every surgical mesh, all must pass LAL testing to prove they won't trigger a deadly immune response. A contaminated implant can cause septic shock, a condition that kills over 250,000 Americans annually. The connection is invisible to most patients, but horseshoe crab blood sits between them and potentially fatal contamination every single day. But this billion-dollar industry depends on a wild animal population, and that raises a question the industry can't ignore. Every spring, horseshoe crabs return to the same beaches to spawn, a ritual unchanged for nearly 500 million years. Marine biologists track population numbers every year using tags, surveys, and trawl data to monitor the health of the species. Conservation groups and the biomedical industry work in an uneasy partnership, both needing the crabs to survive, but for very different reasons. The stakes extend beyond medicine. Migratory shorebirds like the red knot depend on horseshoe crab eggs as critical fuel during their 10,000-mile journey. That is why scientists have spent decades developing a synthetic alternative, one that could end the industry's dependence on wild crabs entirely. Recombinant factor C, known as RFC, replicates the horseshoe crab's clotting reaction using genetically engineered proteins produced in laboratory cell cultures. Instead of bleeding live animals, RFC is manufactured in bioreactors, the same technology used to produce insulin and other recombinant drugs. The European Pharmacopoeia accepted RFC as a valid alternative in 2020, but the FDA still requires traditional LAL for most applications. The regulatory transition moves slowly. Pharmaceutical companies have decades of validated LAL protocols they're reluctant to abandon. For now, horseshoe crab blood remains irreplaceable, and that means the bleeding facilities continue to operate at full capacity every harvest season. Each tiny vial represents a chain that stretches from the ocean floor to operating rooms around the world. The finished LAL ships from distribution centers in insulated containers, maintaining the cold chain every step of the way. Hospitals, pharmaceutical plants, and research laboratories receive fresh LAL shipments on a weekly schedule. The supply must never run out. In hospital pharmacies, compounded medications like chemotherapy drugs receive their own LAL tests, an additional safety layer before they reach the patient's bedside. Only after a clear LAL result does the medication travel to the patient, a process repeated thousands of times per Dalton Y across every major hospital. The patient never sees the test, never knows the name horseshoe crab, but that ancient animal's blood is the reason this medicine is safe to enter their veins. During mass vaccination campaigns, a single facility can administer 10,000 doses per Dalton Y, every one of them LAL certified. The scale of modern pharmaceutical manufacturing is staggering. One production line can fill 50,000 vials per H-hour. Every single one of those vials traces back to a horseshoe crab that bled blue on a stainless steel rack in a coastal laboratory. Each LAL test kit carries a unique batch identifier, linking it to the specific harvest date, bleeding facility, and production lot that created it. The largest LAL producers operate year-round, with harvest season blood-sustaining production through the winter months via massive frozen reserves. Frozen blood reserves stored at -80 degrees Celsius ensure that production never stops, even months after the last crab was bled. Every year, as spring returns to the Atlantic coast, the cycle begins again. Boats heading out at dawn to harvest the world's most valuable blood. The horseshoe crab doesn't know it carries a substance worth more per ounce than any precious metal. It simply follows the same instinct it has for eons. 450 million years of evolution produced a blood chemistry so unique that modern medicine still cannot fully replace it. That blue color, that ancient copper chemistry, stands between humanity and invisible bacterial threats that would otherwise be undetectable. At $60,000 per quart, horseshoe crab blood is among the 5 most expensive liquids on Earth, surpassing gold, scorpion venom, and vintage champagne. This single facility processes blood from over 100,000 crabs per season, transforming it into reagent that protects 1/4 of the global pharmaceutical supply. The Atlantic horseshoe crab, Limulus polyphemus, is the only one of 4 horseshoe crab species used commercially for Lao production. State wildlife officers monitor every harvest, enforcing strict seasonal windows and per-trip limits that protect the breeding population. Population surveys show that careful management has kept Atlantic horseshoe crab numbers stable, a rare success story in commercial wildlife harvesting. Acoustic tags now track individual crabs after release, proving that over 85% survive the bleeding process and return to normal behavior. Back in the laboratory, endotoxin reference standards must be prepared with microgram precision. The accuracy of every test worldwide depends on these calibration materials. The World Health Organization maintains international endotoxin reference standards, ensuring that LAL tests produce consistent results regardless of which country performs them. High-throughput robotic systems now run thousands of LAL tests simultaneously, but the fundamental biology hasn't changed since the first test was developed in 1977. The FDA's bacterial endotoxins test chapter remains one of the most referenced standards in the entire United States pharmacopoeia. Finished product worth millions of dollars sits in climate-controlled vaults. Security is pharmaceutical grade because the contents are irreplaceable on any timeline shorter than a full harvest season. Only a handful of these facilities exist on Earth, and each one operates within a narrow seasonal window dictated by the crabs themselves. Facility managers coordinate harvests months in advance, balancing production demand with conservation quotas and weather conditions. The harvest season spans roughly May through July, 3 months to collect enough blood to supply the world's pharmaceutical industry for an entire year. Peak spawning aligns with full and new moon high tides, and that is exactly when harvesters know the largest concentrations of healthy crabs will be accessible. Before the sun rises, the boats are already on the water. The cycle that has supplied modern medicine for over half a century repeating once more. Every sealed case carries holographic security labels and tamper-evident packaging. Counterfeiting pharmaceutical testing reagents is a federal crime. 48 vials per case, 12 cases per carton, hundreds of cartons per shipment. The numbers multiply rapidly. Air freight carries LAL to pharmaceutical hubs across 6 continents. The temperature monitor inside each container logs conditions every 30 seconds. If the temperature deviates outside the specified range at any point during transit, the entire shipment is destroyed. No exceptions. When a shipment arrives at a pharmaceutical plant in Germany or Japan or Brazil, the first thing checked is the temperature indicator. Green means safe. Red means rejected. Inside facilities like these, Louisiana L testing happens around the clock. Quality control laboratories that never close because production lines never stop. A single large pharmaceutical plant can produce 20 million injectable doses per month, and every production batch requires LAL clearance. That green clearance stamp is the last barrier between the factory and the patient. The moment when horseshoe crab blood officially says this medicine is safe. 45 years after the first commercial LAL test, this tiny vial remains the gold standard for pharmaceutical safety, an ancient solution to a modern problem. From an animal that predates the trees, from blood that runs blue with copper, humanity found its most reliable guardian against invisible microbial threats. 450 million years of survival gave the horseshoe crab a blood chemistry so sophisticated that our best laboratories still study how it works. The technicians who bleed these crabs understand the weight of their work. They handle an animal most people have never seen to protect patients they'll never meet. Every spring, the ancient ritual continues. 500,000 crabs harvested, bled, and returned to the ocean that shaped them over geological time. $60,000 per quart, 15 times the value of gold. Flowing drop by blue drop from an animal that was ancient before the first dinosaur drew breath. This is the Horseshoe Crab Blood Factory, where ancient biology meets modern medicine to protect every injection you'll ever receive. The next time a needle enters your arm, remember, an animal older than the dinosaurs already made sure it is safe. If you found this journey fascinating, you'll want to see how snake venom is extracted and turned into life-saving antivenom. The link is on screen now.